So these are all excellent questions, and I think this is a good time to actually introduce our panel. Just a reminder for those who are listening virtually, you can actually go on to the KDM5C support group Facebook page and ask your own questions, and I will help moderate those questions and ask our panelists, so you'll get answers in real time. So before I introduce our panel, I just wanted to make note, while organizing Rare Disease Day, this morning session of Rare Disease Day, we really wanted to have you pick who you wanted to hear from. And so we posted onto the Facebook group, and many of you indicated that you wanted to hear from a neurodevelopmental pediatrician. So you wanted, because you had questions about behavior and maybe some of the science. You also wanted to hear from an endocrinologist, so we invited a pediatric endocrinologist, and many of you were very interested in maybe starting your own rare disease foundation. So, we have the perfect people to actually talk to you about that, and you probably met them upstairs. So we actually invited Dr. Lisa Shulman, who you just heard from. She's a neurodevelopmental pediatrician, also the interim director of the Rose F. Kennedy Children's Evaluation Rehabilitation Center at Montefiore. And then we invited Dr. Cristina Valianatos, who is currently a genetics education specialist at the Jackson Laboratory. I also wanted to mention that Cristina, Dr. Valianatos, actually did her PhD in Dr. Shigeke Awazi's laboratory at the University of Michigan, and one of her expertises during her PhD was developing a mouse model of KDM5C and looking at different types of outputs. So looking at behavior of these mice, neurodevelopmental abnormalities in the brains of these mice. And so if you have any questions about the actual science, or if you're confused about some of the genetics, she'd be the perfect one to ask. We also invited a pediatric endocrinologist, Dr. Lisa Underland, who comes from us from Montefiore Medical Center. And so if you have any questions about endocrinology or certain hormone questions, maybe you received a diagnosis that involves altered levels of certain hormones, she'd be the perfect one to ask. And we invited some rare disease foundation founders, and I'm actually going to let Dr. Stephen Walkley do the honor of introducing them. If you have any questions about how to start your own rare disease foundation, this would be the perfect opportunity to ask them some questions.
Thank you, Hayden. So I think we're suffering a little bit of confusion here. I guess, Hayden, you would like the folios to be up on the panel now. So Jessica.
I'm going to do clinical questions first and foundation questions next.
I thought we would just have everyone.
Oh, great. Cool.
On the panel.
Okay.
Yeah.
So let me just say a word about Jessica and Mike. We have a program you're hearing two different aspects of the Rose F. Kennedy Center today, the CIRS program that's so critical for clinical care. And the research side is the Intellectual and Developmental Disabilities Research Center that I'm the director of. And, we've been sponsoring Rare Disease Day now for quite a number of years. And one of the programs within this IDDRC, we call Operation IDD Gene Team. And basically, and you'll hear about this afternoon in some detail, because in some ways the three families that'll be speaking and the clinicians and scientists that are part of that all are sort of a manifestation of what this IDD Gene Team concept is all about, where we're trying to put together families with their clinician, with scientists who are studying that condition, and build collaborations around that. And a lot of this starts when a child is diagnosed, has a developmental disorder, and is diagnosed through, for example, whole genome sequencing, which is so common now and revealing more and more diagnoses for rare diseases. And so the folios can give the details of this themselves, but they were involved in this program after their son was diagnosed with a disease called SALA disease, which is a lysosomal disorder, which happens to be what my lab works on. And so we created an IDD gene team with their son's pediatrician, with Ben, with Mike and Jessica, and then with members of my lab as the scientists involved. And I had this tutorial, it was actually back in 2017. And at the time, Sallow disease is exceedingly rare lysosomal disease. Lysosomal diseases are like Tay-Sachs disease, which some of you have heard of. There are about 60 of them. We got together over in the Kennedy Center and did a tutorial, did a lay sort of explanation of what Sallow disease was all about and so forth. And Sallow disease, among these 60 different lysosomal diseases, is so unique that it doesn't fit in with any of the other major categories. So there's no foundation or patient support group for Sallow families. And you know, we said this to Jessica and Mike, and even went so far as to suggest, well, you know, maybe you want to start a program like that. And you know, Jessica, I remember so well, you're looking at me and you said exactly what I think you would expect a parent to say under those circumstances, and that is, can't think about that right now, I just want to take care of my son. And but then 6 weeks later, I hear from Jessica and Mike, and they wanted to start a foundation. And so that was 2 years ago, and that foundation has just grown enormously. And that's, so as a family that has just recently gone through this experience of literally creating a foundation to support families and to support science and research on their condition, they really represented really an ideal example. And so I'll just leave it at that. Jessica and Mike, Olio.
Okay, so why don't we just start the questioning. Feel free to ask anything related, if you had questions about certain slides in the presentation. In fact, Molly, do you want to join us on stage too? Why not? So if you have certain questions about the presentation, feel free to ask Molly or Dr. Schulman or any of our other panelists. Or if you have a question about something that was unrelated to the presentation, feel free to ask. And again, if you're watching virtually, I can help moderate questions as well. So feel free to ask any questions.
Well, my question is for the researchers. Number one, I'm hoping that you're going to expand on the survey work and maybe publish it. Is that your plan? Number two, I just wonder if you are in any networks that might search electronic medical record databases that could dramatically increase the number of diagnoses. It occurred to me when I went to a rare disease conference last week back in Ann Arbor that they mentioned things like ACT network and the TIN network. And then I spoke to the speaker afterwards and I said, wow, our son's KDM5C didn't end up on his problem list in his electronic record until we had a hospitalization and things had gone, you know, sort of south. And yet the diagnosis was back in 2012 or 13. And that if we don't call it a problem and list it there, it won't be searchable. And then my third goal had been, since you're a PI in Spark, to get KDM5C on that list. But then one of the wonderful things about the Facebook page is that after I posted that, I realized that the girls who might not have a diagnosis of autism would never be eligible to participate in Spark. So if you could sort of address that complex group of questions that all relate.
I'll take a stab at it because you and I are definitely on the same path. So I didn't bring it, but I have a prop through our Spark participation. I have a prop through the Spark grant. And by the way, there's like a TED talk on Spark. I don't know if you've come across it, but I have a prop that lists all the genes they've discovered that contribute to autism risk. There's like 150 on there. And the process of families contributing their saliva and the saliva of their child with autism increases that list, and the process is iterative. So when they have a sample and a new gene is hypothesized, they rerun all the samples again. So the very first thing I did when I heard about this event was to look on the list and see, was KDM5C there? And it wasn't. So I was like, wow, well, I'll be really, really curious what percentage of these children meet the criteria for autism. And again, you saw that it was about 25%. But this is where my comment before about the importance of getting appropriate diagnostic expertise involved, because quite a few people whose parents do not report that the children have autism, their parents do report that they have social skills difficulties, that they're overly friendly, that their greatest concerns about the future, which were listed, relate to social. And with the change in the Diagnostic and Statistical Manual to 5, it was designed specifically for this scenario where you have some identified etiology for developmental problems, but the diagnosis of autism, for example, is one that you make separately. Does the child meet the behavioral, social communication deficits and some rigidity and sensory things? And so it is that, as we all hear all the time, autism is a very, very wide spectrum. And I suspected in reading the very limited amount of information we had clinically that there are more children here who would meet ASD criteria. And so that's a network that I have access to. I think it would, the whole purpose of the Spark is, again, to kind of further these connections. It would be a way of generating more cases. There are problems with electronic medical record and in terms of who lists diagnoses and how readily they're available. So the Spark was my main kind of clinical way to greater recognize the mutation, get more information about it clinically, and interest researchers, hopefully here in the IDDRC and elsewhere in this condition, because that connection is made. Other people, I'm sure, like more through the scientific, more bench-related things, also have connections, but it's kind of pulling those things together where I do feel it's kind of at the infancy.
Would you consider going through one of those other databases to pull more cases so that maybe this is a rare disease, but maybe it's not. Maybe it's much more common than we think.
You know, almost all the kids were identified in the past couple of years. There's only 1 or 2 who were identified much longer. I think that we're really at the beginning of the process where whole exome sequencing is available. For most of our families, it's only available through a protocol. And so I think this is just the beginning of there being many, many more children diagnosed with all possible mutations, and KDM5C is one of them. I do think a discussion, and maybe this is meant to take place in kind of clinical genetic forums, about how to make sure these clinical, etiologic diagnoses are best coded, because I know exactly what you mean about, EPIC and the electronic medical record. And it depends how somebody codes it as the primary problem or not. And there's definite work to do. So I do think this is one of those things where it's going to require having all the appropriate players in the right place at the right time. But I do think this is the beginning of that process, because, again, every week we get results back on whole exome sequencing that are new. Again, I haven't had any KDM5Cs, but all of these mutations are being cataloged and in databases. Beginning of what will be routine care, because whole exome sequencing is more readily available. But all of your kids had this level of testing. But We follow 2,000 children at the Kennedy Center with autism. 150 of them have had whole exome sequencing. So again, the things need to catch up. But I think it is important to involve clinical geneticists. Marion in the audience, panelist on how to best or routine as something that we seek to database.
Okay, I have a series of questions. I'm going to try and do it as concise as I can. This is for the one that studies the mice. I'm sure what your name is. Christina. And also the lady that did the survey. There were some things that our daughter has that weren't mentioned, and I wanted to know if you've seen them in the mice and also if there was others in the survey and it just wasn't mentioned, or if it's like her own genetic makeup, because I know that they have their own genetic makeup. So temperature regulation is a huge issue for our daughter, constantly feeling hot. And I heard other kids also mention that on the page. Ketotic hypoglycemia, she had as a child, lots of hospitalizations for it. Carnitine deficiency, Chiari malformation, neuropathy, carpal tunnel. Those are the main ones.
So those are great questions. In my experience, don't know any reports from the literature about the mice or any other model organism for KDM5C that has displayed those symptoms. But you brought up a really great point at the beginning, that is this something from KDM5C or is this just some sort of natural variation? And I think I've seen that in the Facebook group. I'm sure many of you have those types of questions all the time, right? Because each of us, we look different, we act different, we have different preferences, right? And this is all because of very small changes in our DNA. And these are mutations, right? So some mutations are just kind of normal. They make my hair curly, they make you tall, they make different preferences for food or ability to metabolize drugs. And those can be benign or they can have big changes, but kind of not really have any major health consequences, or they can lead to something more serious, right? So there's kind of depends on where that variation lies. So it could lead to something like KDM5C or can just kind of change our skin color, for example. So these types of questions are kind of hard to determine. This is why surveys like this are so important to kind of get as much data from the population as possible. But this is why also you heard everyone talking about the survey saying, well, we only have 16 people or we only have so many girls or whatever, because, if you just took a few of us here on the panel, well, that's just a select few, right? And so most of us are women. So you might say, oh, well, it's this, but actually it's because most of us happen to be women, right? It's not a true sampling of the variation amongst all people. So it's kind of the same thing with that survey, right? So we just need more and more people, more and more research, more and more time to kind of pinpoint, okay, well, if most people have this variation or this phenotype, this problem, maybe that is because of KDM5C, because that's the common problem. But actually, if only like 10%, maybe that's just kind of something that's just an environmental factor or a genetic factor. It's very complicated. But at least those phenotypes, those clinical manifestations that you mentioned, I personally haven't seen it in the mice that I worked with or in any of the literature that I read. But again, it doesn't necessarily mean it could be something less common.
Somebody else was shaking their head. Can I just ask, just because, do other children with KDM5C have temperature?
Yes, I know. I know two children. One is my son, and another one is in charge from Manchester. They can't cope with hot weather. They can't cool down at all.
Okay, so raise your one from each couple whose child has temperature problems. Please raise your. All right, this is very significant, and we will definitely add it. Again, it's like the phenotype. It's not physical. It's not it's not developmental, and it's not behavioral. I guess it is physical in some way. That's very significant. So thank you very much for bringing that up. When I do remember your little responses, because there were so many things that you mentioned that likelihood that these child with this mutation has all of these things, and it's not due to this. But yeah, Most of the other things did not appear in other people's description, although, like we listed, there were small heads, children with large heads. There was somebody who needed surgery for craniosynostosis. There was plagiocephaly. So there were a number of brain abnormalities.
Well, and I wonder with the neuropathy and carpal tunnel.
Right.
If there are other children, they just haven't had an EMG because so many of them complained of problems with hand function and fine motor. So, it might just be that they don't know yet. I don't know.
We also lumped those under physical problems. About that, they seemed possibly tonally related. So, yeah, you're asking excellent questions, and we would love more information. And a second survey or instrument. I mean, one of the things that we were thinking of was we really want to know about the autism. Okay, I'm going to close. Yes, I'm very interested in autism. But I also do feel, reading the description, that we want to know specifically, are the social weaknesses at the level of meeting criteria? And because that would allow us to go in a certain direction clinically and get this listed as a particular problem associated with autism and be on the list of things that are looked for. So if we were to send through a link, there's a parent instrument that's the most general to help us answer that question. We're hoping that you can take up your time to allow us to answer that question.
And I'll just add that, so for our survey, as you saw, when we asked those open-ended questions, it's just kind of how you interpret the question versus when we ask those very specific. We didn't know to ask about temperature regulation because it's not really in the literature anywhere. So it probably, if we had asked that question specifically, like with that short stature question where we asked open-endedly, 6 people said it. When we asked directly, 13 said it. So when we learn more and more about some of these things, we can, produce some additional screening tools or use ones that are already out there, asking these questions to better understand what actually is out there. So hopefully the more we learn, the more we can get more information from all of you.
I can also add something to this, and maybe Dr. Underland can also add this. But so KDM5C is expressed in many places. One of those places is the brain. And in terms of temperature regulation, there's a region of your brain called the hypothalamus. And that is really important in temperature regulation. And this hypothalamus, I believe maybe Dr. Vallinalis can help me. And is KDM5C highly expressed there? Do you know?
I don't know the distinct brain region expressions. I remember looking at, and it's at least pretty consistently on working across the brain. I don't know if it's
right.
higher in the hypothalamus.
Right. So I mean, there are many. This isn't a be-all, end-all or an explanation, but it's a possibility maybe that maybe expression in these temperature regulating regions might affect thermoregulation. So that could be one possible explanation. Do you know anything, Dr. Underland?
I don't know anything directly about the thermoregulation. We do have some patients with associated hormone problems that have problems with hormonal regulation, with thermoregulation, but there's other patients who don't seem to have hormone problems that have problems with thermoregulation. And generally, connections between, if you have one hormone problem, often you have other ones too. So it could be related, it could not be related.
It also brings to mind kind of autonomic.
I was going to mention is that because of the large number of individuals on psychotropic meds, the relationship could be psychotropic meds versus the disorder itself. And
I'll just reiterate the question. It's about a lot of the people are on psychotropic medication, which could have effects on this as well, right? Because of the medication.
I have a question for the endocrinologist. Should our children be seeing an endocrinologist to look for other things? And then our son had a major regression associated around the time of puberty, which is delayed. And we've had so many different opinions, and Dr. Schulman might comment too, is to whether there is a regression or a change when children with developmental disabilities in general go through puberty or autism, and what to expect, and is there any way to prepare parents, because we didn't feel very well prepared for sure, and just to comment on that and sort of how to address those issues.
Yeah, I mean, in general, I always encourage people to seek subspecialty care if there's a question. If your pediatrician is saying, yes, you have a child who's kind of growing on the same percentiles, they seem to be matching what we expect for the parent height, the puberty seems to be in normal timing, the question of whether you need to bring in an endocrinologist is always, that may depend on availability. I definitely think if you have somebody who's not on the charts or kind of falling off the growth curve, then I usually encourage people to see an endocrinologist, even if it's just for monitoring. As far as hormone changes during puberty, I've not really heard as much of a description of regression, but there's definitely a lot of behavioral changes that parents deal with when patients undergo puberty. There is a certain amount of, I think, media attention on testosterone and how that really makes your child have a lot of behavioral changes. And I think in my experience, it's made some parents very afraid of testosterone and very afraid of puberty. And ultimately, if patients aren't making enough testosterone, they're afraid of supplementation. So we need, if your child is not making the hormones they need, and, it's beyond just being delayed, they're just not making them, I think appropriate supplementation is very important for bones and things like that. As far as how to prepare, it's always a difficult question to kind of to tell parents exactly what to do, because every child is going to be different, and some are going to have it's going to be a much bigger response than others.
I'll second all of that, and that the teenage years, just like we were saying, every child with any mutation or rare disease is going to have all the regular teenage stuff too. Is in terms of regression, if there were a frank regression, or there is a frank regression in physical skills, loss of self-care skills, loss of language, that always requires medical evaluation. That's highly unusual. When we get into more kind of maladaptive, non-compliant behaviors, and so that I would look at more educationally, the school setting, the supports. So the distinction between a frank regression and having more difficulties, I would deal with separately. But a frank regression is something that we take very seriously. Medically, it's not expected.
So what about you? And then the other microphone's over here. Yeah.
Our daughter is 4, and she starts off with anxiety, but then it turns into angerment. I just didn't know, will she grow out of that without a prescription, or?
You know what, when we looked at the age range of families who responded on the survey, I was thinking that there will be families whose children are very young here, and you may get information that could be quite scary, and your child may not follow. So I want to first acknowledge that. The second piece to that is I would want so much, there are so many strategies to manage anxiety and aggression. So this is going to fall to me in the category of we are nowhere talking medication for this 4-year-old, but rather making sure that her her preschool program and therapeutic program is giving her a means of communicating her anxiety and strategies for addressing her aggression. So kind of optimizing her communication and giving her the vocabulary to speak her emotions and frustrations is something that I hope her educational program addresses. And if it doesn't, then going back to your school district and with documentation that this can be associated with a condition can sometimes help you in advocacy.
So this is maybe I'm not particularly qualified, and I'm not saying this with authority, I'm saying this as a question. It seems like creeping around on the Facebook page, and it's happening maybe perhaps again here a little bit, which is with a very small sample size, and with what I feel a deep sense of camaraderie with you all, deeply. Like, you're amazing. I do get a little bit nervous the degree to which, I mean, even when we raise hands, like I was wondering, like, wait a second, if we did this with my other child, or any child, I mean, I've taught middle school for a while in a couple of different age groups. Like, I get a little bit nervous, not that there's not the best of intention, because I'm doing this too right now, but I would love for you to speak a little bit to the uncertainty of what we're talking about. Because I'm really, whether it's reading about vitamins and that kind of stuff, or pharmaceuticals, like it just seems like our sample size is so small. And we care so much that we hear like, wait, maybe that was better. And man, when we do nothing different, 1 week is amazing. And we're like, we've done it, we're there. And if we took the wrong medication that week, or the right one, we never know what to attribute it to. And it just makes me a little bit nervous when we're all here raising hands and thinking, because I get excited and then I'm like, wait a second. And could you all talk just a little bit as authorities with some meaningful research experience to say like, hey, what does an N of 16 really mean? And like, how careful should we be? I don't think we'll be totally depressed. I just love to know how much are you in the back of your mind saying, yes, but. I'd love to hear a couple of those yes, buts too, just because I don't know when you're saying, yes, do that.
So real fast, just about little statistics, N of 16, what does that mean? If this was N of 16 genetically identical mice, I would be like, that's an awesome sample size because they've been in the same environment, they are genetically identical, I did these tests, and 16 is enough to have, even if you have a couple, random fluxes, that you have like a really good number, that you should have like a really tight range of variation. 16 humans from around the world, from different backgrounds, from different origins, right? Different environments, different ages. That's a huge variation. That is not enough. That's not enough to draw any sort of conclusions. And that's why kind of every other sentence ended with, but we only have 16 people, right, for the survey. So we can make associations and we can say this is likely, and we can draw on the other information that we know from mice, from flies, from other literature, from other people, from things that we know how the gene works, from things how we know how the brain works, or other autism, intellectual disability disorders. But we cannot make any definitive conclusions, right? So that's a great thing to always keep in the back of your head.
Maybe this is a good point. I know Dr. Julie Seacombe, you had your hand raised a moment ago, but either you or Hayden, I know this afternoon you're going to speak about not the phenotypic variability, but the function of the KDM5C gene and protein, and maybe what generates so much phenotypic variability. You want to say just a couple of sentences on that now to help us all understand? Or Hayden?
Sure. You can. Yeah.
So I would also reiterate that, it's complicated, right? Humans are complicated because they have other genetic things going on in the background. It's also true that it's very unlikely that all of your kids have the same mutation, right? So different mutations, as I'll talk about this afternoon, don't necessarily cause the same things to happen, right? So I think that the N of 16 is nowhere near enough. And in fact, I actually had a question for you guys, which is, KDM5 is part of a family, and there are also mutations that occur in KDM5B, which is actually very similar to KDM5C. And I wondered if anyone had reached out to the KDM5C group with their kids that had been affected by KDM5B. because this could be a way to increase that sample size by saying, you know, these genes aren't necessarily doing exactly the same thing, but maybe they do some things in common. That no one's reached out to the Facebook group with?
I don't think so. I actually, when I was, you know, when I first entered, when I first was welcomed into the KDM5C support group, I also searched on Facebook for a KDM5B support group that was similar, and I didn't quite find one with such a rich and, you know, active community. I know that there's another group, I think it's called a KDM5B variant group or something, but there really wasn't much discussion there. I know, you know, from the literature, there are, you know, plenty, I forget the exact number to date, but there are plenty of mutations or plenty of alleles of variants of KDM5B that produce very similar phenotypes. And what's very interesting is that many of those individuals present with autism spectrum disorder as well. Actually, I think a higher percentage compared to our very small sample size here. But it would be very interesting, I agree, to maybe, you know, look further to see if there's some sort of support group for KDM5B, KDM5A. There's a KDM5D, but there have been no affected individuals for that.
Yeah, so there are 4 KDM5 families, like genes, like Kaden said, A, B, C, and D. C and D are on the X and Y chromosomes, respectively. And then kind of the function of what this KDM5 family of genes does in the body. There's a whole separate family of genes that do basically the opposite function. And so these are the KMT2 family. So that has A, B, C, D, F, and G. Don't ask me what happened to E. But these guys also are linked to neurodevelopmental problems. So that was also some of the work that I did. Some of the work that Shigeki Iwase's lab does at the University of Michigan is to look at this kind of opposite family of genes also, because they're kind of working on the same thing, just in an opposite way. And so it's just really interesting how these, even though they're distinct genes, that if you lose the function, if you have some sort of problem, it could lead to the same phenotype. So maybe that's some information that we could provide after the event, and we can start searching to see any sort of similarities in these other genes, if there are any similarities in the clinical manifestations.
I would also like to reiterate that, you know, mutate, like these changes that you guys have all found in your families in KDM5C, it's new, right? You know, I started my lab here at Einstein 11 years ago, and at that time, I think there was probably half a dozen, maybe, families with KDM5C that were around. And so this explosion of new like changes that have been seen in families, like we're really just starting to catch up with like what changes that happen do they cause in neurons. And so we're, as you guys are learning, we're also trying to learn as well.
And 20 years ago, we didn't even know what this gene did. So it's a very new field in terms of learning exactly what KDM5C does in relation to the very complex brain that we have, right? How it grows, how it functions, how it develops. So it was about 15 years ago when we first discovered, okay, this is KDM5C, it's a gene, this is how it functions in the brain. And then it was like, oh, there are some people that have mutations in this gene, and it leads to this type of clinical outcome. So it's just under 20 years, so it's a very new field, but it's, you know, groups like this that can help to progress the research and discover more.
And just, I guess, adding on to that, I know that Dr. Julie Seacombe is going to talk in the afternoon about, you know, what KDM5C actually does in certain cells, but it, you know, is one of those important links between DNA and protein, which actually does a certain function. So if you have, you know, certain different variants of KDM5, when they're affected, they can lead to disruption of the expression of certain proteins. So Dr. Seacombe is going to talk more about that this afternoon.
I have a question, or actually I have a few questions. One is going back to the autism diagnosis. When Gabby had a full neuropsychiatrist workup, it came up that she had some autistic tendencies, but they ruled out autism. Now I'm wondering, should we be revisiting that perhaps? Would be interested to know on that. Also wanted to know, gene therapy is a big thing you hear about in the news, and wanted to know, is that something that's possible for these kids? And if so, what does that look like? Or is that so far off that it's not realistic to consider at this time? And then I'm also eager to hear from Jess and Mike about starting a foundation, as I know many of us have discussed that and want to do that and want to know where to start. So eager to hear about that too.
I'll take the first question, which is that I think it is worthwhile to go to an autism center or somebody with specific autism expertise. There are gold standard instruments, something called the Autism Diagnostic Observation Schedule, that is an important piece of data for kind of borderline calls. That can open the door to irrelevant. I'm going to hook on to that, the idea that really all of my responses, as opposed to being KDM5C specific, are developmental guidance. So although KDM5C is new and there are only 16 responses in the survey, I'm giving these responses based on years and years of evidence-based.
I'll take the gene therapy question, but I just have kind of a follow-up question to can you talk a little bit about autism manifestation in males versus females? Because there's a little bit of a difference, is there not? And then the second question is autism diagnosis in the US versus other countries, because I know we have a lot of families that are not in the US system.
That's true, actually, because I think we come from the UK, and I think that the criteria for autism in our country is actually different than UK. So perhaps we should, agree on what we consider autism all across the globe, because otherwise it's kind of meaningless.
And that might be a bigger fish to fry in terms of making the whole global medical community agree on a diagnosis criteria. However, I think it's just important to acknowledge that there are differences in criteria, not necessarily that just because there's such a wide spectrum, right?
I mean, when you were talking before, I recognize what you said about my son, and he's actually yet not fully diagnosed. He said he fulfills some of ASD criteria, but not all of them. And he goes to an ABA school.
That I don't consider myself an expert. I mean, outside the United States, they'll use more ICE, which are a bit of a throwback more, where there's still Asperger's, there's still West, but idea is that there are social and communication deficits. All three are required. That there are 2 out of 4 required in the domain of response. But they can be, common refrain is if you know 2 children with autism, you know 2 children with autism, that no 2 are the same. But what I will say is when people come here to train, specialists, the most common thing that they say that the mild end of the spectrum is something that they would never recognize previously. And the point of making the diagnosis, again, not talking KDM5C specific, we're talking developmental here, that for higher functioning children, they can be taught social skills through social curricula. Generally, to gain access to that curriculum, you have to name that that's a need. There are social weaknesses. Children who have more cognitive impairment than ABA methodology is generally a helpful methodology. Here in the United States, it's impossible to get ABA without an autism diagnosis. So again, clinically speaking, clarifying if a child meets criteria for autism is very helpful in terms of pulling together an appropriate intervention package. And it does require expertise. Go places where there was genetics expertise to get this diagnosis, and not everybody has access to that. You have to go to a place that has autism expertise in order to get an autism diagnosis. That diagnosis is important in terms of prognosis, in terms of current. And I say this to rooms of people, children, we don't yet know what their mutations are. This is just a finding related to developmentally delayed. The major risk factor for warranting a diagnosis of autism spectrum disorder is among the group of children who have developmental delays. Common thing we do here is when there are children who have various syndromes, various significant delays, and the question that the families are asking is, does he or she also have autism? So it's something to know yes or no to. Craft an appropriate intervention plan that will address all of the child's needs. Okay, you're on.
So I don't even know kind of where to begin with how we started our foundation for solid disease. Like most of you, if I had these doctors in the room, I wouldn't be asking about how to start a foundation. I'd want to know exactly what's going on with my child. So I'm here to answer any questions that you want regarding starting a foundation. But our foundation started 2 years ago in September of 2018, just like you are doing right now here. Dr. Walkley had amassed a team of doctors with an idea of what solid disease is, the best that he could think of. And we met in Terrytown. And so you guys are off to such a great start. The doctors had a think tank. And then I set up therapy sessions for families. So families could go from vision therapy to occupational therapy to speech therapy to physical therapy. So families went from therapy session to therapy session while the doctors met. Like you, when we got our diagnosis, there was nothing. We had to have Dr. Walkley call another doctor in California to even put us in touch with another. It's like, how are we living in 2017? Like, how is this even possible? So I guess we've had to file so much paperwork, and I don't even know where to start about how our story, our foundation's just going gangbusters right now, and I don't know why. I don't know how God has intervened in the entire way. When we got our diagnosis, we happened to have a man in the room who runs foundations for a living. His name is David Helper, and he runs Israel Peace Forum. And that's all he does, and he's our best friend. And he, 15 years ago, took a job working for a man named Jonathan Jacoby, whose son had something called Niemann-Pick. And 15 years later now, because of Dr. Walkley, there is not a cure, a treatment for Niemann-Pick. But David, 15 years ago, took a job for Jonathan Jacoby and watched him start a foundation. And here, David was in the room with us when we got our diagnosis from a different institution, but he immediately put us in touch with Dr. Walkley and Dr. Wasserstein. And this is how we started. And the entire time that we have started this foundation, we say, hey, David, what paperwork do we need to file? I'll do it, just tell me what I need to do. So David would say, you have to file this paperwork, this corporation letter, and we just did the grunt work. We just went by his word, and we just did what he told us to do. When I approach families now, we have just grown gangbusters. There's a first diagnosed case in Latin America, and they happen to be very affluent people there. And they're having a huge concert where they've already sold 4,000 tickets and raised $60,000. And this event is happening on Saturday. Their daughter has solid disease. The mom was a novela star, and the dad is a politician, and they have gone gangbusters. But when I address my families, first and foremost, I address them as pioneers, because I don't say we're going to have a cure, because I don't know that that's the reason. But I always say to the families, you have a purpose, and your child has a purpose to pave the way for future children. And if that is something that touches you or is going to motivate you in the depression that you feel often when you have this sort of diagnosis, because it's depressing and it's scary, I just say Ben's little life has a purpose, and that has given me so much. I think it's really helped us. And the relationship that you guys have with each other is so important. It's so important to lift each other up no matter what you're facing. And I know you want a cure right now, but unfortunately, that when they started cancer research, there wasn't chemotherapy immediately. There wasn't radiation immediately. But my Ben and our efforts are going to eventually come up with something. And that's a scary thing, too, to think about. It might not be in time for your child, or you might not see the benefit. But if you make the benefit for other people in the future, that's something huge. That it gives you power, I guess. So anyway, there's a lot to how you start a foundation. If you have specific questions, we're happy to expand. But now we have, I don't know if you want the results so far. We started 2 years ago, and now we have a full-time hired a technician working at the National Institutes of Health. We have cell models, we have mouse models, and then at the last symposium, there's work going on with Costa de Brenes here with knockout mice. We're working on the knock-in mice at the NIH, so they'll be sharing information. And then you all talked about gene therapy. So at the World Symposium, which now we've been going to for the past 2 years, a group from CHOC, Children's Hospital of Orange County, approached us, and they're young investigators. They had won a big award for their gene editing therapies. They approached us on if we would like to collaborate with them. They have won $8 million for rare disease. They're not going to give us the $8 million, but some of it will go towards us. And I had to ask my team, I had to be very, I had to ask the board because now we have a board of directors. I had to ask the doctors because I don't want to step on anybody's feet along the way. And now we just announced that we will be collaborating with this team who will be doing gene editing. And the NIH is going to ship the little mice over via FedEx to Orange County, and they're also going to share. It's going to expedite their research, and it is going to save them money. I mean, they did get a big grant, but all the money needs to go to expediting and getting this research going. So, I'm sorry, Mike. Go ahead. Do you have any?
A tough act to follow. One, thank you for hosting us, having us here today. I'm amazed at you guys getting together like this. I found, we were thinking about this actually this week where, what's the biggest things that our group has accomplished? And I think one of the biggest things is having family networks, having that support group there where you're not alone. You have other people you can talk to. Hey, I see this with my child. Do you see that? That's really important. That will help you guys kind of get through this stuff or just cope with it. I think that's huge. To your other point too, I think we've talked about genetic exome sequencing. I know for our case, we were first diagnosed as cerebral palsy, and we kept digging, and through genetic exome sequencing, we found the exact cause of what it was. I'm quite sure there are people with SAL out there that are misdiagnosed, and I'm quite sure that's the case for what your children have. And I'm sure, through genetic exome sequencing, you're going to find more cases, and it gets cheaper to do this test. You will find more too. To start a foundation, it's kind of simple. It's not that hard. You really have to, depending on your state, and I'll just be brief about this, you have to first find the name for yourself for your corporation. Then you have to basically incorporate yourself, so you have to write up articles of incorporation. Once you've done that, then you can just file with IRS for 501c3 status, and you're a tax-exempt nonprofit group.
Make sure you register as a public charity. We've made the mistake of registering as a private charity, and so Amazon Smiles, Facebook, all of these things that collect money, you cannot, if you are listed as a private charity, thank you. You cannot list yourself on Amazon, Facebook, get bigger donations from, there's a Jewish benevolent fund. Anyway, so it's really important to file as a public. And we felt so small, like, whoa, of course we're private, but no, you have to be public. So just make sure in the eyes of the IRS you're that.
What about with these international families? Because, how is that working with
Right.
this family in Latin America? Because I know we have families from the UK that want to start a foundation as well. Do we start two separate, or do we work together and have a UK and a America branch? Or?
The question would be, is where do you want to do your research?
Correct.
Where you're going to collect all this data, you're going to decide what to research. Where do you want that to happen? You have to figure out who's going to be doing that, who's going to be collecting this data, and what you're going to be doing with it. So that's a key question there. Because it could be multiple locations, it could be one. We have one here in Rose F. Kennedy Center where we have our knockout mouse, but we also have our knock-in mice in NIH. So we have to fund, certain times both places. So it's a question of, what you guys want to do. I know it's the beginning phase, it's a little daunting, but you have to think about that. Europe's tougher for regulations, especially if you want to collect
IRB reports.
patient data collection. That's what we're kind of interested in. And if there's other individuals affected, that's a different language. That could be a hurdle. I know I'm getting ahead of myself with this, but you have to think about translation if you're doing that in the States. So.
Because most people speak English.
Hold on.
Oh.
So you think.
And most people in Europe actually speak
Okay.
languages, I speak four languages, I'm Italian myself, and that's kind of the norm.
Yeah. I guess I'll make this announcement to Dr. Walkley. So we all think that ours is a Scandinavian disease. It stems really from Finland and the Vikings. But we now have actually two diagnosed cases in the Dominican Republic, and it's supposed to be Scandinavian. How are they? And now yesterday, because of our foundation, I got a call from Guatemala. And there's a case now in Guatemala, and the mom doesn't speak English at all. And so now this is coming up, the Dominican Republic, this family that's gone gangbusters. I put them in touch because I don't speak Spanish. And this is so Guatemala, like from Scandinavia to Guatemala. So there's going to start to pop up. And so we are starting now STAR chapters. Now Norway, we just got together at the World Symposium. She's so excited, and she now has filed the paperwork in Norway, which went way faster than the United States. So she is a sister chapter in Norway. And now I'm going to ask the Dominican family, who's having this huge party on Saturday, now that there's more Latina cases popping up, I'm going to ask them to start a chapter in Latin America. So then we can, and then at the World Symposium, there were also more doctors in Mexico thinking that there were many cases in Mexico that they have just misdiagnosed. So we all speak, that's going to be a barrier, that's going to be a barrier for us. It's the language barrier, really. So I don't know if that answers your question.
So I actually just wanted to get in one question from the Facebook group. And I don't know who the appropriate person to answer this, so I'll just let you speak. But someone said, I am concerned about how to tell if a toddler behavior issue is just something that's a normal tantrum or part of this disease as far as how to handle these important problems, if that makes sense.
I can only give an opinion that I would treat it as a regular toddler problem, but also make sure that the child's intervention is focusing on communication. And make sure the child isn't also sick with regular childhood illness like ear infection that would make them more likely to be irritable.
Can I just answer the gene therapy question from before? So the short answer is it's still a far ways off. For those of you that don't know, gene therapy would be delivering back into your body a gene that is lost or not functioning well. And so you may have heard this even just in the news on NPR or something. There's a woman recently that got treated for a sickle cell disorder. And so the way that this works can be various different ways, but again, kind of delivering a faulty gene back into your body. So there are two things for KDM5C. One is that it's primarily a brain disorder. So how do you deliver something to the brain and where and when, as opposed to something like sickle cell that's primarily a blood disorder that you can kind of do a transfusion. I'm very much simplifying this, right? But you can kind of do a transfusion and it gets exactly where it needs to go. As opposed to the brain, it's just very hard to deliver something like a gene. And again, where do you inject it into the brain? You know, you can't really deliver a gene in a pill. There's so many different parts of the brain, how the cells talk and connect. It's quite complicated. So that's one thing. The other thing is that at least from the research, we've realized that KDM5C is a developmental problem, that it's not something like an Alzheimer's or a Parkinson's that kind of manifests later on and causes you to degenerate. It's something that happens very early on, maybe even earlier than we can tell, and the symptoms are happening very early in development, right? So when is the exact therapeutic window? When could we deliver a treatment if we have a treatment? That's a very big unknown question. So work in the Iwase lab that I did for my thesis research, I had a kind of a therapeutic model essentially. And so this was in in the mice. So as was mentioned, like huge steps are just having mice, having cells, having research. And so we have those labs like the CCOM lab, labs like the Iwase lab. Research is actively happening. So for a lot of rare diseases, that doesn't even happen because there are just too few people to kind of care about that disorder, right? So this is a huge step. So don't be discouraged. Progress is happening. But it's just a matter of time. So some of the work that I did was, I had mentioned KDM5C kind of has this sister family of genes that do the opposite function in the body. And so it was having that KDM5C mutation, losing function in KDM5C, but also losing function in the other guy as well, one of the other genes. The thought was to kind of balance so that these two genes work in opposition. So if you lose one, you're kind of shifting the balance of what's supposed to happen. But if you also lose the other one, maybe you can kind of neutralize and bring things back to normal. And for a lot of things in the mice, we looked at the neurons, we looked at different genes, we looked at behavior. Overall, it was very, very favorable. A lot of good things were happening in the mice that had these double mutation. But again, it's mice, it's early. These were kind of genetic knockouts, so it'd be different to have to develop like a drug or a therapeutic or something. This happened from very early on in development, so whether or not we could do this type of therapy later on, when that therapeutic window would be. These are all questions, and now we have some foundational research. We can do more research, we can ask more questions. So it's something that's being worked on, it's just a little far off.
Hayden, if I could just make a comment, in terms of accelerating the science, one thing that you may not be aware of is that NIH has what are called meeting grants. They're R13 grants, and where a scientist who's working on like a problem like KDM5C would put in a grant to sponsor a meeting that would bring together scientists focused on these kinds of issues that you're hearing here. In rare disease, these meeting grants are coming largely out of the Office of Rare Disease at NIH. And so they're interested in the kinds of conditions like is represented in your families. So it's another mechanism. It's not what Mike and Jessica used for their initial meeting. That was a much more focused think tank. But the meeting that we're having at NIH in September of this year is using an NIH funding mechanism. It's an internal mechanism, but they have these external R13 mechanisms as well. All you need is to team up with some scientists to help you organize that meeting and to submit the grant. But it can be very, very helpful for diseases like this, conditions like this.
So yeah, in the back.
I don't have a question, I just had a comment, and it's kind of spurred by this gentleman's comment about the sample size, and then the developmental pediatrician's comments about making sure you get the right services. So we're kind of at a different point in our journey with our son as he's a bit older. But I guess if it helps anybody, we got through our journey from experts who didn't know anything about KDM5C, but it was having the right developmental pediatrician, the right geneticist, the right neurologist, the right lawyers, the right social workers, and where he is now at his group home. None of them know anything about KDM5C. But we've kind of made it through like the bad stuff, I guess. So take light in that. There's a lot of people out there that will help you, and they don't have to know about KDM5C. They just have to really care. So
I think you bring up a really great point too. We have 2 boys in our audience today that are 18. So all the children will always be your children, right? But they won't be recognized as children by the medical system forever. They won't be out of the school system soon and out of that support network. So, to the clinicians on our panel, what can families do when kids turn 18? What can they do to prepare, to expect? Of course, not for KDM5C specifically, but for these developmental disabilities.
So I just wanted to leave enough time for lunch and, to allow you to meet each other. The afternoon session is at 2 o'clock, but we'll start heading down around 1:45. The front 2 rows will be reserved for you. So feel free to sit in the front, and if there are no seats, you can find a seat in the audience. We also have a break or in another room in the building where it'll be broadcast to. What room was that? I think it's the third floor, 351. Oh, it's 451. Okay. So the front 2 rows will be reserved for you, and 451, this will be broadcast to. So lunch will be on the 5th floor again. Your children are waiting for you. And I bet they're also very hungry. So thank you. If you have questions, feel free to come up and ask the panelists.